Welcome again, everyone. My name is Abdellaoui. I work at the Amsterdam University Medical Center, even though it says everywhere I'm working at the Vrije Universiteit. At the Department of Psychiatry. So I am actually a geneticist. I have a genetics background. And I like to look at the intersection of behavioral genetics and population genetics, and I like to look at larger patterns of variation and look at how genetic variation behaves geographically. So basically, I like looking at how genes influence behavior, but also how behavior influences the genetic makeup of a population. And then you have to think about things like migration and partner choice. And in those studies, I keep running into socioeconomic factors being the biggest drivers. So that's why I keep running into economists and social science geneticists. And that's why I am here now. And I'm going to tell you something about genetics. It's going to be a historical overview, and I'm going to try to convey some of the concepts that you are going to be working with in the coming week and the coming years. So how many, I have the feeling that most of you have like a social science economics background, right? How many people have like a biology or a genetics background? Let's see a couple of hands. Economics? That's the majority, yeah. Okay. So I'm going to talk about genetics. Genetics is basically, I have to point this at something.
Don't click again with the mouse on the slides.
Oh yeah, thanks, that works, yeah. So in genetics, we try to ask the question, where do similarities and differences between living organisms come from? It took quite a while before we got close to the answer of that. So the first recorded thoughts about this were from Pythagoras and Aristotle in 350 BC. So Pythagoras, he thought that the father supplied the essential characteristics of a person, so the form or the idea behind who you are, and that the mother supplied the material building blocks. And Aristotle, he was convinced that semen was purified blood and that mixed with menstrual bloods to make children. That's where sayings like your bloodline or it's in your blood, that's where that comes from. And then, so those were the prevailing ideas for a long time. And then in the 16th century, somewhere in France, I think some medical and professionals were developing the concept of biological heredity in diseases using actual legal concepts of inheritance and property and wealth. So that was interesting because now we're back to using those same concepts to try to explain things like inheritance of property and wealth in social science genetics. And in the 17th century, scientists showed that in animals, like breeds like, and that all female organisms produce eggs. And the microscope was invented. And that helped scientists see sperm cells for the first time. And they claimed that they saw little human beings in those sperm cells. And they also believed that these human beings, that they also have sperm cells that also contain human beings. And those human beings also have sperm cells. And that everyone that's ever going to exist already existed at that point in time. And that's called pre-formation, is that idea. And then about a century later, I think maybe the most important insight in the whole field was was described by Charles Darwin in his theory of evolution. He had a theory where there are inherited pieces of information were passed down to the offspring, and that's information of how a creature looks like, and it can change sometimes a little bit because of mutations. Those mutations are then selected by natural selection, and that's how you get differences between individuals and eventually different species. So this was his first drawing of a so-called tree of life. So he has different subspecies here that he called D, B, C, and A. And he said here, I think, I don't remember exactly what it said here, but he was contemplating that if these two groups didn't intermix for long enough, then they would diverge from each other and become new species. And that's actually what happened here in this modern drawing of the tree of life. And this is a similar tree, but this is based on genomic clustering analysis. So then in 1865, this German monk who was playing around with peas in his garden, he found out the actual laws of how these pieces of information that Charles Darwin talked about, how those are passed on and how they are expressed in living beings. So there are 3 laws of Mendel. Does anyone know them by heart? It's a lot to ask, maybe. Okay, so the first one is the law of segregation. So we each inherit 2 alleles for every genetic variant, 1 allele from father and 1 allele from the mother. And of those 2 alleles, we pass down 1 randomly again to each child. So that's the first law. And the second law is the law of dominance. So some alleles are dominant and some are recessive. And an organism with at least 1 dominant allele will display the effect of that dominant allele. So in this example, the dominant allele is the big A, and the dominant phenotype. Can anyone see what the dominant phenotype here is? These are P's, by the way. These are nice. Yeah. So smooth surface, that's the dominant phenotype. And the wrinkled surface, that's the encoded by the small a, and that's, you need 2 of those in order to express that trait. And then the third law is the law of independent assortment. And that's law says that genes for different traits are passed down independent from each other. So a gene for whether the P is smooth or wrinkled is not passed down together with, let's say, a gene that determines the size of the P or the color of the P. Those are passed down independently from each other. So this law is actually not always true. And the reason why not is going to be very important for the stuff that I'm going to talk about later, for things like the GWAS design, that a lot of that is based on why this law is not true. And it's we don't pass down genes individually together. We pass down whole chunks of our DNA. And if genes occur together on a chunk that's passed down, then they get passed down together and they correlate in the population, because the closer they are to each other, the more likely they are to be passed down together. And if they correlate with each other, if they occur together in the population, that's something that we call that linkage disequilibrium. So they are in linkage disequilibrium, and that's an important concept for the development of GWAS later on. And by the way, I want you to feel free to interrupt me with questions anytime you don't understand something, because I have an hour and a half apparently, and I don't know if I'm going to fill all of that by myself. Yeah, yes.
So the law of dominance, I mean, of course it's true, though. My feeling, and so that's why I'm asking the question, is that it's actually fewer phenotypes who follow dominance, while most of them have a linear type of effect. Is that correct?
That is correct. For the traits that we are interested in, which are called complex traits, so we're going a little ahead of ourselves, but that's fine. Complex traits are influenced by many genes with very small effects. Those are most of the traits we are interested in. And GWASs now are showing us, at least the results are very strongly suggestive for the fact that dominance plays a very small role in these complex traits. Most of those traits are, for the vast majority, determined by very small additive effects, but we will get to that later.
So yeah. Just Mendel just stumbled upon one that had dominance.
Well, there are a lot, like there are traits, usually and they often call those Mendelian traits also, so traits that are influenced by a small number of genes or even one gene. Those are can have dominance effect, like whether you have blue or brown eyes, for example, or there's something like Huntington's disease or sickle cell disease is also a recessive trait. So those traits do exist, but they seem to be more often traits that are influenced by either one gene or a few genes with big effects. Yeah. Okay, so in 1875, Francis Galton, this is Charles Darwin's cousin. He came up with the idea to look at twins. These are not the classical twin studies as we know them, because back then they did not know about identical and fraternal twins. He just was trying to study whether he could change twins by changing their environments. But he did, I think, suspect the existence of a special type of twins. It says here, there are twins of the same sex, so alike in body and mind, that not even their own mothers can distinguish them. Their features, voice, and expressions are similar. They see things in the same light, and their ideas follow the same laws of association. So he was talking about identical twins here, which we will come back to later. And Francis Galton, you could say that he started to get a little interested in social science genetics here. This book is called Hereditary Genius. And he applied statistics to show that offsprings of eminent figures, so people that did well in society or that are extremely talented, were more likely to succeed in high-profile professions. Yeah, so he was objectively a very smart guy. He invented a lot of the things that we still use to this day, things like correlation and linear regression. He invented that. But yeah, like morally, ethically, I don't think he would have survived today, but it was a very different world back then as well. So here you see a family tree here, for example. This is Galton's side of the family. This is Darwin's side of the family. This is Wedgwood's side of the family. And he labeled, Wedgwood is a famous businessman that was also related to them. And he labeled people in the family as either brilliant scientific ability or normal. As you can see, it's only the males that are labeled. But it was a different world, and he was also, well, he was a huge racist, and he also did some other things that had horrible consequences, like inventing eugenics. So he saw that, yeah, so things like intelligence and how well people did in society, that was heritable, and that gave him the idea to improve the genetic quality of the population through selective parenthood. So actually, this is an old idea that I think Plato also suggested something like this. But he gave it the name eugenics, and he made it really mainstream. So it was a very popular movement back then, like the medical authorities, all the scientists, the political spectrum, the entire, whether you were left or right, everyone thought this was a great idea back then. And yeah, that led to horrible atrocities, things like forced sterilizations for marginalized groups of the population. And then during the Second World War, it got linked to race, and then it took genocidal turns. And after that, eugenics was considered uniformly bad by everyone. But I think we also have like an ethics talk later this afternoon, and I assume that there's going to be more talks about this as well. Because basically all the bad stuff that happened was because they took an incomplete understanding of genetics and then went very hard into applying that social policies with very little compassion for the people involved. So we have to keep stuff like that in mind when we try to link our genetic findings to things like social policy. We try to be very careful about that. Ronald Fisher, also a very brilliant man, also a huge proponent of eugenics. He and also a pioneer in the field of genetics. He did something very important. Back then, it wasn't really clear how quantitative traits behaved. So things that you could measure on a continuous scale, things like your height or your intelligence or your blood pressure, your BMI, those are quantitative continuous traits. And those were hard to reconcile with those laws of Mendel, because Mendel's laws were governing very qualitative traits. So whether it was wrinkled or smooth, whether it was yellow or green. And then Mendel, he found out that actually also for quantitative traits, these genetic variants follow the same Mendelian laws. So let's take this example here. Let's suppose that we're looking at height, all right? And if you look at the upper one here, you have 3 different genotypes. So you each person, as we said, inherits 1 allele from each parent. That could be either A1 or A2. So you could either be homozygous for A1, then you get 0 centimeters added to your height. You could be heterozygous. If you have 1 of A2, then you get 1 centimeter added. And if you're homozygous for A2, you get 2 centimeters added. So for every A2 allele you carry, there is 1 centimeter added. That's the Mendelian trait here. And below that, you have 2 genes that can add a centimeter on height. You have the A gene and the B gene, and A2 adds 1 centimeter and B2 adds 1 centimeter. And suddenly, you have 5 different phenotypic outcomes. Either you're homozygous for A1 and B1, and you get 0 centimeter. And there are 2 ways of carrying 1 extra centimeter. Either you have A2 or B2. There are 3 ways to get 2 centimeter added, and etc. And here we have 3 different genes. And the more genes you add, the more it looks like just the normal distribution. So that's how so-called complex traits work. And that's most of the traits that we are interested in. They're influenced by many genes with small effects, usually additive effects. So this is an additive effect because there's each 2 allele just adds 1 centimeter. And even disease traits that look like they are qualitative, because either you have diabetes or you don't, either you are schizophrenic or you're not. But below that binary outcome is a continuous, what they call a liability scale. So how much risk do you have to develop the disease? So there is a normal distribution there, and once you cross a certain threshold and you're in an environment that triggers the disease, then you'll get the disease. So even binary traits, we treat those as complex traits with an underlying normal distribution. Okay, so in the 1920s, the first classical twin study was conducted. And with classical twin study, I mean a twin study where they compare identical twins or monozygotic twins with dizygotic twins, where identical twins share 100% of their DNA, their genetic clones. And dizygotic twins share just as much as a normal brother or sister. We say they share half of their DNA. They actually share more than 99% like all we do, but there's a part of the DNA that varies in the population, and of that part, they share half. And almost all of the traits that we looked at, so this is a meta-analysis of twin studies from the last 50 years. Identical twins, so this red distribution, they show a higher correlation than fraternal twins. And when that happens, you say there is probably a heritable component to the trait. Here are the types of studies that have been studied in twin studies. Psychiatric disorders have received by far the most attention. Those are, I think, the most mysterious diseases, especially from a biological point of view, but also quite heritable. We'll get to that later. So almost all of the traits that were looked at show some heritable components. And that's why this first law of behavior genetics was coined. All human behavioral traits are heritable. So and of course they differ in how heritable they are. So this was from an older meta-analysis of twin studies, and the purple bar is the heritability, the genetic influences. So heritability means so if something is 50% heritable, like let's say testosterone here or a heart rate, it doesn't mean that 50% of the trait is caused by genetics. It means that 50% of the individual differences in the population can be attributed to genetic differences in that population. So it's a relative measure. It can differ in between populations where you look at, and all of the traits here have a heritable component. There is one exception here, and that is religion. That shows 0% heritability, and that's not whether you're religious or not. That's which religion you have. So whether you're Christian or Muslim or Protestant or Catholic, that's all determined by the family that you are born in. But whether you're religious or not, by the way, is quite a heritable trait. Okay, and then another very big discovery. I'm not sure which one is bigger, the Darwin's theory of evolution or this double helix, but they're very related. They basically found out the molecular mechanism behind evolution. They found, yeah.
Yeah, being religious is quite heritable, but which religion is not that heritable?
Yeah.
Studies looking at overlap between genetics of being religious and other behavioral or mental health traits.
No, but I
like,
would. Yeah.
It's like, I don't know, maybe if you are into compulsiveness or neuroticism or things that I'm not insinuating anything, but there's of course a machinery of those traits and development.
Yeah. That would be, I would be very interested in looking at that, but there are not any GWAS as far as I know on whether people are religious or not. So this is all based on twin studies. Twin studies might have looked at that, but I'm not even sure that those exist. But if anyone is interested in starting a big GWAS consortium of whether people are religious, I would be interested in doing that. Yeah. Yeah, that means that 50% of the differences in the population are because of genetic differences in the population. So then that can differ for between populations. For height, for example, how tall people are, that's 80% heritable if you look in a Western society. But if you look at developing countries or poor countries, you see a much lower heritability because everyone in that developed country, in Europe, for example, most people have an okay access to food, to diet. But in a poor country, there's a lot more variation in whether people get to eat enough or not. So there you get a lower heritability because the environmental differences explain more of the variation, explain more of the individual differences between people. What the definition of a population?
Yeah, I guess that's the bias explaining.
Yeah.
I will need to define my population first.
Yeah.
And is it enough if I just talk about large European?
Well, if you can be more specific, if you can say, for example, this is the Dutch population, or well, actually, it's just in the data set that you're looking at. It's not even sure how generalizable those, because even the data sets that we study, they have a bias. They are not representative always of the entire population. And there's even for things like educational attainment, they saw that even within the US, the heritability difference, whether you look at poorer part of the population or a rich part of the population, because of the environmental differences that influence whether people get to express their genetic potential. So it's very relative. And when I say population, actually, it's like just the data set that you're looking at. Yeah. Yeah, that matters. Mm-hmm. Yeah, yeah, yeah. Any other questions about the heritability part? Okay, so this is a very famous paper by Watson and Craig. Actually, so this is a book that talks about overlooked contribution of this woman, Rosalind Franklin, who her observations played a key role in discovering this major finding. And it is the molecular mechanism of how evolution works. So you have two helices that are connected to each other in such a way that if you split those two helices up, you have exactly the same information in both of those helices. And the A always binds to the T and the C always to the G. So if you split them up and you throw in a bunch of loose ACs, T's and G's, then you get two other double helices formed, and those are almost always perfect copies of each other. Almost always, because sometimes you have a small mutation, and that gives natural selection something to work on. So that was very big, and they discovered how, shortly thereafter, how these different combinations of nucleotides code for amino acids, and amino acids are the building blocks of proteins, and proteins are building blocks of all the living creatures on this planet. So then we can start. So from this point on, we can start thinking about trying to figure out what's explaining the heritability. What is which genes or genetic variants are responsible for these individual differences. So the first type of study was the so-called linkage study. And in a linkage study, they pick a certain genetic variant. They could not measure the whole genome yet in the 1980s. They pick a genetic variant that's like tags a very big region. When I say tag, I say it correlates with a lot of genetic variants in that region, because remember, we pass down big chunks of DNA and not separate genes. And this is a family tree where they try to find out whether this genetic variant is inherited together with a certain disease that they are interested in. So this is, I think, the first successful linkage study where they discovered the gene that codes for Huntington's disease. So these black circles are individuals with Huntington's disease. Circles are women, squares are males. And they discovered a variant on chromosome 4. If you carry 2 of those, so that's a recessive variant, then you will certainly develop this horrible disease somewhere in your adulthood. But these studies only work for very big effects like this. So either Mendelian disorders, which are disorders that you get 100% if you carry the gene, or just genes with very big effects like the Alzheimer's gene. Alzheimer's is a complex trait, but there's one gene called the APOA gene that has a huge effect. Or diabetes is a complex trait, but you have the insulin gene that has a big effect. Those were found with linkage studies, but they weren't very successful in finding genes for complex traits. So for a long time, for complex traits, they tried to find genes based on a priori hypotheses that they had about the genetic variant or about the disease. For depression, for example, major depressive disorder, they knew that drugs that did something with that serotonin transporter had an effect on the disease. And so they hypothesized that the gene that encodes for the serotonin transporter must also then influence major depression, right? And there were many studies published, some of them very high impact, like this science study here from 2003, where they found all kinds of interaction effects on between the serotonin transporter and major depression. So here, for example, they show in this figure a very strong effect, interaction effect. Now, people that carry the short version of the allele and experience severe maltreatment have a higher chance to develop depression. So the effect of childhood trauma depends on your genotype for that serotonin transporter, is what they found. And there are a lot, thousands of these kinds of studies that were done in relatively small data sets, hundreds or maybe thousands. Who used to would be a big candidate gene study, usually it's hundreds of people. And then this is a recent, much bigger study. This is from 2017, where they actually had like 30,000 people genotyped on that specific serotonin transporter gene. And I found none of these effects. They could replicate none of these. So these were all false positive findings. So the whole entire literature, there's a whole field that's just completely consists of false positives. Titus mentioned that already, that some journals now say we don't even accept these candidate gene studies anymore. Unfortunately, I still see them published sometimes. I even see them get grants sometimes. Here's another example. The same team, by the way, also in Science in 2002, found a similar interaction effect with the monoamine oxidase gene. So that's a gene that was hypothesized to have an effect on aggressive behavior. And it went so far even that in here, this is a news article from Nature where about a court in Italy that reduced the sentence of someone that murdered somebody because he carried two of these variants. So these false positives, they don't just ruin the literature for us, they can have actual real life consequences. So it turns out we don't understand the genome well enough to think of a priori hypotheses that work, I think is the conclusion here, and also that you need much bigger data sets to detect small PET-CSP as well. Oh, sorry. Yeah, you go first, Peter.
I know.
Yeah.
So Kathy and Moffat were also part of the meta-analysis paper, though, right?
They were? Not,
I think so, no?
let me see. Oh, I can see that. I know the last author. I'm not sure because I saw that he still kind of believed in his old
Okay.
stuff.
Yeah.
But I could be mistaken. Yeah.
Blame them in 2003 to try to do this deep. Yeah, I think it was a revolutionary study, and they did try. I mean, you know, not sure it's for science, but it was something very innovative at that time. So.
Yeah, I do wonder, like, could that be us as well? Like, yeah, I don't know. If it could also be like just publication bias, because if you have 100 or 1,000 groups doing an experiment, just by chance, some of those are going to find an effect. Yeah. And those are going to be the ones that are going to be happy and send it to science.
Yeah, that was kind of my question too. Okay. Like, what is law? Nothing law, because it just said if you look for no candidate, you will 5% of the time you will find it.
Yeah. Yeah. What was behind all that? Like the psychology field or social science field had a similar thing, right? That when most of the studies that they did turned out to all be false positives as well, and a lot of the big hypotheses that were making people famous. I don't know, is it all publication bias? Is it people wrangling their data until they find something?
Yeah, there are more general effects in the social science than also in economics. That basically too often we also study that about genetics, where this would happen.
Yeah, it was a general trend, definitely.
So just again, because I think we're, you know, I'm doing a lot of debugging work. I mean, I understand how they got there in some sense. They had a theory-based model. They had a theory that was like biologically based, that had serotonin idea. They had a whole bunch of things that were pointing in that direction. And again, I don't necessarily fault them in 2002 for having done that type of work. There were a lot of hypotheses in the literature that were pointing towards precisely this direction. And the idea that GBA could exist, and could be exactly for that particular genotype and so on and so forth. I think they, you know,
a lot of this stuff was.
I don't think we fully understood why the 5-HTT or the NOA gene are not robust. Like, we didn't fully understand why they are false positives, or did we? Maybe I'm wrong, but we just found that statistically they're not. It doesn't hold to the test.
It's just not a real signal that they publish.
Yeah, exactly.
And it gets a lot. It got replicated a lot, but those replications were also false.
We're also false positives. Yeah. Exactly.
How does that happen? I don't know.
Yeah, that's publication bias for sure. Yeah.
But in that processing, shouldn't there be like diversity checks where you, even if it's a small sample, but like 3 independent small samples, and then if the effect happens, for example, you wear 3 and a fold it. I don't know, I expect to do that.
Yeah, so yes, replication should be required, and in big enough data sets like that. But the GWAS seems to have solved a lot of that now, because those things get replicated everywhere. You look, if you have a powerful polygenic score, it will predict the trait in every data set that you look at. So, yeah, so those are convincing, but I'm getting there. Yeah, so one of the problems was that we could not scan the entire genome. So that's, yeah, so it was kind of all they could do is pick a small number of genes to look at. And they had no idea how big these effects were, so that they couldn't really do a well-informed power analysis. So we first need to know what the entire human genome looks like. So the Human Genome Project was, of course, a major step towards that goal. It took a very long time to fully sequence the first human genome, and it cost a lot of money, $3 billion. Titus already talked about how it went. It became. Now I think you said it is like $600 per genome. So that means that in like 15 or 20 years, it got more than 3 million times cheaper. And I don't think there's anything that got that much cheaper that quick. Yeah, you also showed Bill Clinton, that picture of him. What he was saying is, we are here to celebrate the completion of the first survey of the entire human genome. Without a doubt, this is the most important, most wondrous map ever produced by humankind. It was a great map, but still not enough to do the kinds of studies we do today, because they had the genome of 1 or 2 people fully sequenced. But in order to do the kind of studies we do today, you need to know about how those genetic variants appear in the population, and specifically which variants appear together. And I was talking about earlier with Mendel, the third law of independent assortment, that we don't pass down single genes, we pass down whole chunks of DNA. And a chunk like that with a lot of different variants that are correlated with each other or in linkage disequilibrium, we call that a haplotype map. So here's an example of a haplotype. Sorry, yeah, we call that a haplotype, not haplotype map. So each row is an individual, and we're looking at these 3 SNPs. So at this SNP, people can have either a G or an A allele. And this is a chunk of DNA that gets passed down together. So you can actually only have these 3 combinations of these 3 variants. And why is that important for the kinds of studies we do today, you think? Why does that make doing a GWAS easier to know which SNPs occur together in the population? Exactly, yeah. He said you can impute the variants that you have not measured. So if you know that this person has a G here and a T here, you don't need to measure the A. You can guess the A or impute the A. And if you don't have to fully sequence an entire genome, that is a lot cheaper. And money matters when you need very big data sets. So there are a couple of very big projects like the HapMap project and 1,000 Genomes projects, where they took samples from different populations all across the world to map out what those haplotypes look like in a population. And based on those haplotypes, they could design what they call a microarray chip, where you don't need to measure 3 billion variants to capture a lot of genetic variants. 500,000 or 1 million are enough to capture more than 90% of genetic variation in a certain population. And that makes it possible for us to conduct a so-called genome-wide association study. So I think, yeah, the question.
Are they the same for most of it? So are they the same location on the genome, or
yeah?
Different?
So between populations, they are very similar, but there are small differences. But like Yeah, like the breakpoints of the haplotypes are quite similar between populations, but they differ in frequencies between the population. And I think there are small differences, but not that many. Yeah. The genome-wide association study. Okay, so that's where the next revolution began. We were able to test every genetic variant across the genome that we could measure to see if it was associated with a trait. In 2005, the first one was conducted on age-related macular degeneration. So that's sort of a disease that develops at an older age. And I found the first hit on chromosome 1 that Tita showed that nice animation of all the GWAS findings. This is a similar type of animation, but so on the y-axis, on the x-axis here, we have all the chromosomes and the genome-wide significant hits, and these are all the types of traits. So if you see, there was 2007, 8, 2009, 10, 2011, 12, 13. So each year, more findings, and there is this big tower on chromosome 6. Anyone have any idea why it is? So there is a region on chromosome 6 called the MHC region, major histocompatibility complex. And that region plays a very big role in our immune system. And it also shows most variation in our genome because it has to evolve to keep up with all the pathogens it's protecting us from. I think they saw least variation in this region in Native Americans because they were not living with all the livestock that Europeans were living with, so they were therefore less protected to all the diseases that Europeans brought with them. And it seems to be associated with a lot of traits, especially a lot of disease traits, because we like to do these studies on disease outcomes. And the major factor that determines whether a GWAS has any signal or not is the sample size. And that is because we found out over time that these complex traits that we are interested in, the genes are not, the genetic effects are not as big as we suspected during those candidate gene study era. The genes have a super tiny effect, and also because we are scanning the entire genome, so we're doing millions of tests, so we also have that multiple testing burden to deal with. You need a huge amount of people to have a genetic effect reach the significance, the very stringent significance threshold of 10 to the -8. Okay, so we arrived at GWAS. I think this is a good time to have a 5-minute break, 10-minute or 10-minute break, 10-minute break. We have a 10-minute break, and I heard from Pietro that there is coffee there and cookies in the room next to us, so we don't have to go all the way. Sorry? Not anymore? Oh, okay. Okay, but there are nice windows there tonight. Okay, there are some, there are some cookies here. All right, see you in 10 minutes.
Then very only have the coffee and we don't have it in the morning before starting, but we have it at 8:30. And I brought some of the things, and there's some drinks. And for lunch, yeah, everything. We need to go to the restaurant. We have to have our gadgets, and with the budget, that is actually important. I think so, yeah. You have to pay for your own lifetime, that's you have that with you. I can see that should say where we're at. And we can all go together. Right up when you come from, yeah.
Everybody has it.
You want to use the next? Okay. And for internet, they also only the password that is there, but it's also for yeah, there's a view guest. With the password, yeah, which. Okay. Yeah. So with this password, we can connect to the guest. Huh. There's no board.
The thing I'm uploading there, I probably there's only a screen there.
You didn't share the slide?
No, I think it's actually slides, I think, so I thought that, that's what I think. We'll see.
We'll see exactly. And it will be safe on your.
Yeah, I hope we go.
On the laptop.
Yeah. That's so how it's like I used to do that, yeah. But I think the file and you go right there.
Exactly. I think we should break it up.
Stop.
Before lunch. Stop and do another one. So like, Monday morning and.
Yeah.
What do we do there?
I don't know, we'll see, right? We can think about what we want to do with it.
Okay.
Yeah.
Because if we are going to be now,
Yeah.
because the speakers, I mean, I guess, well,
We'll see.
because the camera was on the speakers, right? So all the speakers were. If we're not even online.
It's a really good occasion. It's, like that.
Nicola.
Yeah, it's probably.
I don't think it's around university and nobody noticed it. So. It's part of the university. Is it teaching? No, the university is on the other side of the main line. The university of Venice, couple of days. Well, the different department. But they have like locations,
It's a more actually at the department long time ago.
Yeah,
It's at the border, no?
it is a word. Yeah, it's pretty good. And I think this is more of a like affiliated something. Where a lot of people do conferences and stuff like that. So there's a guy who's in charge of how are the rooms?
Nice. Yeah.
Okay. Okay.
Are you staying here as well?
Yeah, I'm staying until Friday.
Yeah, I live on my lady. Well,
Yeah, I think a lot of people are leaving on Friday after the Friday. How is it to get here from the airport?
it's fine. I have to stay. It's a while with the boat, but yeah,
Okay.
it's yeah. It wasn't too much easy to figure out where I needed to go.
Yeah, I asked someone, monthly figure,
Yeah, so. Yeah,
but it's both. But
So the ride itself, we're just waiting.
right, no, both in.
Okay.
And so,
Yeah, this one is all about you think about it's 35 minutes or something. And then you where the boat over here comes.
Yeah.
And that's only every half hour, so I have to wait for a while.
Yeah, so I did this here.
Yeah.
And there, like I arrived and I had to wait for 25 minutes. I probably just missed.
yeah, they get the schedule.
Yeah. Yeah.
And tonight, I think we're having dinner in the main city, so we have to get both together and we're gonna understand a bit more.
Yeah. have such trouble with eating the goes over the water, I think. Because it's actually cool enough to get to that. To feed.
Yeah, I asked them and they just said, yeah, just go going to the answer. No, sorry, not my. And then walk to the island. Oh, there's water. And not water. So, yeah, it's not very reliable. I was told that particular small, on the fancy beach, right? Yeah. You can get, so, we're going to get lost. And I think that is going to be fun.
Yeah.
Because if you don't have to be the pilot, it's very, very fun. So we know a bit more of the location, I'm the tourist. So, they look down on me.
They do.
I think so, yeah. There's many people who can live.
The real tenant, the
Yeah, many people are there, we most of them live, in the main, it's mostly tourists and Airbnb, hotels and Airbnb and like, yeah, so it's. Most, basically like, no institute. Well, I mean. You have to.
Yeah, also, yeah.
So like, and there's a lot of food bridges that are with staffs.
Yeah.
So carrying groceries is a thing that you literally have to carry them all the time. Right, so if you're an old lady, it's the best.
Yeah.
I knew a couple of friends of mine went to school here, university here, and they lived on the island. But they told me that, you had to walk 20 minutes to the closest to a grocery store that was not outrageously expensive. Yeah, there's like 2 or 3 supermarkets in the city. And they're not super close. They're usually a bit on the end with that and the prices are a little bit markup. So whenever they go off order, they need to compare mostly by person. They need to pay someone to carry all the stuff that is being done.
That's more expensive.
It's always more expensive. And so, yeah, so this friend of mine is living in a carpet apartment that was working days, like during the winter, is terrible. And he had to walk a lot, carry the groceries all the time. It's a bit of a but for a second house, or like you come here every once in a while, it's not really but yeah,
the nicest fallacy.
we call it like a part of the I am yeah.
Okay, well.
So, yeah. I'll make go.
I thought they were just.
I thought you were saying. Okay, we're gonna start again soon, everyone. Okay, so a general announcement. So we're going to have lunch at the restaurant, but they're only going to serve you lunch if you carry the bag. So make sure Make sure to get your badges for the people that haven't already. Yes, so badges are necessary to get lunch at the restaurant. Was there any other announcement, Pietro? Anything else?
I will complete the reference, so just follow me afterwards. And if you don't have internet, there is a guest password somewhere here, so you can get the. If Eduroam doesn't work for you, you can get a password there.
Thank you. Okay, so GWASs seem to produce robust and replicable associations between genetic variants and complex traits. And when they saw that it worked, you can see in the last 5 years, sample sizes have more than tripled. A lot of this is because these big biobanks that Titus already mentioned, UK Biobank being the most significant one, because of its size, but also because of its accessibility. If any scientist can apply for UK Biobank, and they usually give you permission to work with the data. But before these big biobanks, the only way to reach these huge sample sizes was through big consortia. So like the Social Science Genetics Association Consortium or Psychiatric Genomics Consortium is a very successful consortium where just all these different institutes all across the world do their own GWAS in their relatively small data set, and they then throw all the data sets together to one or a couple of analysts that then do the meta-analysis and then compare with each other whether they did a good job or not. And a lot of significant genetic variants were found. So these Titus already showed a lot of those social science results, but I just wanted to show this schizophrenia GWAS, because this was one of the first ones that one of the first diseases, psychiatric or behavioral outcomes that reached that found SNPs that reached genome-wide significance. And this biggest hit here, that was also on chromosome 6, people first thought that it had maybe something to do with the immune system because it was near that MHC region. But it turned out to be a variant. So not the actual the SNP variant, but it was what they call a structural variant. So the structural variant differs in the number of copies it has, but that's something that we don't measure in these GWASs. But it was tagged by those SNPs because it was near those SNPs. So you can still get a signal even though you don't measure it. And it influences a certain type of synaptic pruning, so the elimination of connections between neurons, and a specific type that only occurs, or that occurs mostly in adolescence when people have their first psychotic break. So this was big news because it's actually quite rare to get a new biological insight from a single genetic variant. Like that was the dream in the beginning. We're going to learn a lot about these new biological processes that we don't know about, but it turned out to be extremely difficult. So schizophrenia is one of the few examples where that happened. This is a summary I made of all the traits from the Psychiatric Genomics Consortium. So you have all these Manhattan plots here with all their hits. And here you have the estimation of the twin heritability. And then in the light blue, that's the heritability that's captured with these GWAS signals. So that's still a long way to go before reaching the heritability as expected from twin studies. We're not sure entirely if that's ever going to happen, if those estimates are accurate. But we do know that there's a lot of variation that we don't capture with those microarrays. And we see that especially for schizophrenia and autism, where those rare variants that we don't capture have a much bigger effect than the common variants. And the reason is because natural selection can't weed them all out of the population. Because if it does have like such a big deleterious effect, the natural selection is going to weed most of it out, and so either make it rare or keep it rare, and that's why we don't measure it in these big chips. Yes.
Yeah. Where the common variant, rare variant, or
yeah. Yeah, so in these GWASs, we scan SNPs that are relatively easy to measure. And those are only SNPs that are common in the population because those appear on those haplotypes, and you need to tell the difference between, let's say, whether the person has an A or a G. But if in a big data set of, I don't know, 5 or 10,000 people, only 2 people carry the G allele, then those genotype calls cannot be made easily. Also, our statistical tests are not devised to deal with such rare variants. So testing whether rare variants are associated with a disease is a whole different branch of the sport, if you will. So we can't do the regular statistical test. So people then, for example, go look for the rare variant burden. So how many rare variants does one carry in total that either appear in a certain gene or just across the genome? Because many of these variants only occur maybe once in one person or in one family or in a couple of families. So yeah, that's a different part of the field that I'm not going to talk about, but is very important and is getting more important as because these sequence data sets, for these you do need whole genome sequence data sets. These are growing. A big part of UK Biobank is now has been sequenced. So and we know that a lot of the missing heritability is there. So it's going to get more important in the field. But there's still a lot to do with common variants. So looking at these single genetic variants and trying to find out things about the biology has turned out very difficult. But what I also like to do is take the aggregate of these genetic effects together, because one genetic variant explains very little, but if you take all of them, then suddenly you have a substantial amount that you can do something with. For example, look at genetic correlations between traits. So for psychiatric disorders, they saw that a lot of the genetic risk is shared between disorders. So one of the most blue spots here is between bipolar disorder and schizophrenia, which means higher genetic risk for bipolar disorder gives you a higher risk for schizophrenia as well. And then here on the right, I have some traits that are interesting for this crowd, so intelligence, education, attainment, and income. Most of them show a negative genetic correlation with psychiatric disorders, because the higher genetic risk for psychiatric disorder is generally not going to help you in school. But there are a couple of interesting exceptions, namely autism shows a positive genetic correlation, bipolar disorder, and anorexia. So we're not exactly sure yet why this is. There's a polygenic score study that helped us get a clue where we looked at the polygenic risk score for schizophrenia and bipolar disorder and saw whether it predicted whether people got a creative job later or not. And it did in the Netherlands, in Iceland, and in Norway it was replicated. So this is from an old hypothesis that a higher genetic risk for especially bipolar disorder that's more often seen in artists and people that have creative jobs. So polygenic scores are a very useful way to look at the aggregate of the signal, because a single genetic variant does not have a lot of predictive power. But if you sum all of the alleles that someone carries and weigh them by the genetic effects that's estimated in a GWAS, suddenly the score becomes predictive. And it's even, you don't even have to limit yourself to only the genetic effects that are significant in the GWAS, because we're missing a lot of real signal because we're being so stringent with that multiple testing burden. So if you just take, the more SNPs we take, even if you take all of the SNPs, the more predictive the polygenic score seems to get. So you use the modern polygenic score analysis usually does include all of the SNPs of the entire chip and just weigh them with the genetic effects as estimated in a GWAS. Especially if you're talking about behavioral traits, those are highly polygenic. And the bigger the GWAS gets, the more precise these estimates get and the more predictive your polygenic score gets. So I think height has made the most progress so far. So this is a distribution of height polygenic score across different GWASs. ranging from, so this was 130K individuals, this is 250K, 700K, and this is the most recent one. The biggest GWAS done so far, 5.4 million individuals. And then this is also the first complex trait where they have found all of the SNPs that explain that SNP heritability that they estimate from a GWAS. There were about 12,000 SNPs. So you could say that height is the first trait, the first complex trait that's now completed with the GWAS. There's no more SNPs to be discovered. And so but that's just for the common variants. It's not the entire heritability yet, because there's a lot of rare variants still that we haven't found yet. So these are 12 polygenic score groups, and here, so this is the lowest polygenic score group. This is the highest, and here you see the differences between the lowest and the highest. Those are these blue dots here. So this goes from 0 to 12, and then the newest one goes from 0 to 22. So the difference between the lowest and the biggest one is 22 centimeters. But I think another way to quantify how predictive these polygenic scores can get is by looking at the standard deviation of the outcome around the prediction. So if you predict something, the standard deviation around it is the standard deviation of the phenotype, which is this one, the y, times the square root of 1 minus r squared. And r squared is the explained variance of your polygenic score in the phenotype. So the upper bound, the maximum predictive power that you can reach, is the standard deviation of the phenotype times the square root of 1 minus the heritability of the trait. So that's never going to be 1. So these polygenic scores are not diagnostic. They always have some noise in it. And this, you can compute what the noise is if you, let's say, we take height. Height has a heritability of 0.8 in a lot of the populations we look at. It has a standard deviation of about 6.5 centimeters. And that means that the standard deviation around the prediction is about 3 centimeters. So the 95% confidence interval of a prediction done on a perfect polygenic score for height is about 12 centimeters. So that's about this much. That's about the 95% confidence interval that you can reach if you have the perfect polygenic score, if you have all the genetic effects estimated. But that's height. Height with a very physical trait. We're looking at a very different animal of traits, behavioral traits, and especially socioeconomic outcomes. They behave quite differently. So Titus already talked about these GWASs. These shown here are the ones on educational attainment and income. So those are the main measures of socioeconomic status in modern societies. And what is being captured with these GWASs is the big question. And by the way, the genetic correlation between these two, between educational attainment and income, is more than 0.9 in most of the populations we look at. So we're looking at almost the same signal, which is, I think, just a proxy for socioeconomic status. And there's not a gene that codes for your high school diploma or for how much money you make. These genes, they influence very low-level biological processes that influence things in your organs, and they influence all kinds of traits. Those traits influence each other. Those traits influence also which environment you get exposed to. That environment influences you again, and influences also all those processes inside you. And all of that big mess of processes all gets captured in this Manhattan plot here, in these GWASs. So it says here, the quote from this review paper we wrote: when the nature of the predictive ability of polygenic scores for behavioral traits is not fully understood, applying them in either scientific research or the clinic can lead to incorrect interpretations and conclusions. Who has seen this movie? It's shown this movie a lot in ethics class. So this is a movie from '97. The term polygenic spore did not exist yet, but it's actually about polygenic spores, where people are only allowed to be born when they have been screened for their genetic risk for a certain disease. They grossly overestimate the heritability of these traits in this movie. But the actual process is not science fiction anymore. There are now actual companies that are offering this. There's people who undergo an IVF procedure. They have to choose an embryo anyway, say the people that run these companies. So you might as well, and they have to look at the genetics of those embryos anyway. So it's not really a big step for them to also make polygenic spores for a bunch of traits.
Sorry, but
Yeah.
they just need to look at chromosomal abnormality, right?
Yeah.
So they don't sequence the embryo.
No, but they already extracted the DNA.
Yeah.
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 So it's not more invasive for them to make a polygenic score. Barely, no. Yeah. So that worries scientists a little bit. I think there may be some regulation in Europe, but I'm not sure. In the US, it's not on the radar of.
It depends from country to country. I think that Netherlands has a relatively open regulation in that regard, but there are other countries in Europe that have more stringent regulation based on previous laws. I don't know if there's any law that targets polygenic scoring for embryos.
Yeah.
But there are other previous laws about selecting embryos that have been applied to this.
Yeah.
And the US does not have one.
No.
Yeah.
Yeah, so these are US-based companies. And this cute baby, this is the first baby that was born out of such a procedure. Her name is Aurea Jen-Mais-Mikrotsky. And it says here, Aurea doesn't know it yet, but her birth was very special. She is the world's first PGT baby, meaning she is statistically less likely than the rest of us to develop a genetic disease or disorder throughout her life. So this is from the frequently asked question website, portion of the website from this company. It says, does genomic prediction screen purely cosmetic traits? No, we only provide risk scores for polygenic traits related to diseases, not for purely cosmetic traits such as hair color and eye color. Our goal is to provide improved health to IVF families. And then the question, does genomic prediction clinical laboratory screen embryos for increased intelligence? And they say no. So I downloaded the GWASs of all the traits that they screen. So that's these traits here. They have brain disorder, heart disease, cancers, and inflammatory bowel disease, and type 1 diabetes. And I computed the genetic correlation with all these non-disease traits. These are just normal dimensions of variation. So anthropomorphic traits, things like height and BMI, reproductive traits, substance use, some psychological outcomes, and educational attainment and IQ. And of course, the genetic correlations are widespread. So an empty box means not significant. If it's not empty, it's significant. And so also so they are in fact selecting for IQ. And not even always in the same direction. See here, this is a positive association. And of course for educational attainment. So the message here is it's very difficult to select for one trait without selecting for the others. In farming, they know this. They try to make tomatoes that are bigger and redder. And those, after a couple of generations, those tomatoes started to lose their taste because the same genes that determine the size and the color also have an influence on the taste. It turned out it's very unpredictable what the genes are doing. So there's a lot of what we call pleiotropy going on. So genes that do influence different biological processes. But there's also something else going on, especially when you are talking about social outcomes. And that's that these GWASs are capturing environmental effects. So I have 5 minutes left. I hope I can cover the whole gene environment correlation topic because that's what the rest of the slides is about. So we've known for a while that there are gene environment correlations on a family level. So we get the first author of this paper to give a lecture tomorrow I hope he's going to talk about this as well. Basically, what they did is they made polygenic scores based on alleles that parents pass down to their children and based on alleles that they don't pass down. So that's the non-transmitted alleles. And they saw that those non-transmitted alleles also predict outcomes in the offspring, especially for educational attainment, they saw this. And that educational attainment polygenic score, both transmitted and non-transmitted, did not only predict educational attainment in the offspring phenotype, but also a range of other health outcomes. And that's because these polygenic scores predict also the family environment. So your parents, if they have a higher education, they're more likely to pass down genes associated with that, but they will also have more resources to provide a more nurturing environment for you. And those things correlate, they're different to tease apart. Here in this study, they saw that a polygenic score has twice the predictive power in a biological offspring than in adopted offspring, because it predicts both the biology and the family environment. So one way to solve this is to do a within sibling GWAS. So that's what they did here for a lot of traits. So this is the SNP heritability of all these traits where they did a within sibling GWAS. Very big standard errors because it's hard to get big sample sizes that just consist of siblings. But the biggest difference was seen in the heritability of educational attainment, which decreased by like 70%. And for all the other traits, they saw sometimes a decrease in their genetic correlation with educational attainment. So that's, they saw if you control for that family environment, you remove the educational attainment signal from other non-education traits, or just health traits, because that family environment, if you grow up in a family with higher educated parents, you're more likely to grow up in a healthier environment and have fewer of these disease outcomes. Okay, and then we got the suspicion that these things might extend beyond the family. So this is the study that Titus shortly showed from me, where we made polygenic scores in UK Biobank, 500,000 people, all of European descent, by the way. So everything that I say here is based on people that have been filtered for their ancestry because of false positives that we wanted to avoid due to population stratification. You hear about that, I think, tomorrow in the GWAS course. But I was curious to see the geographic distribution of all of these more than 30 traits. So these are behavioral outcomes, physical, mental health, things like height and BMI. We try to get as many traits as possible. And this is a measure of geographic clustering. So it varies between 0 and 1, 0 being totally random, 1 being perfectly geographically clustered. And before controlling for ancestry differences, so those are captured by so-called principal components. Principal components summarize just the biggest patterns of genetic variation. And if you plot those on a map, then you get these nice distributions that often separate Wales, England, and Scotland from each other, because these ancestry differences usually show either cultural or geographic barriers. And before controlling for this, a lot of these polygenic scores showed significant regional differences. And if you plot them on a map, then they look a little bit like this. So we were not exactly sure why that was. We didn't pursue it a lot because we were not really interested in ancestry differences, but there are a couple of possible explanations. One, either there is an actual difference between these different ancestries within Great Britain, but it could also be, for example, for bipolar disorder, we saw exactly this map. It resembled it very strongly. And we know that in the bipolar GWAS, a lot of the bipolar cases came from Bristol, which is here in Wales. And if you don't control for population stratification and you have cases coming from a different region, then you don't only get signals from the genes that influence bipolar, but also the genes that show you the difference between a Welsh ancestry and the rest of the country. But so we tried to control for this. So we controlled for 100 species, and then everything dropped except for this big outlier, and that was at the educational attainment polygenic score. That showed very big regional differences. And if we plot them on a map, then we get the same map as just socioeconomic status in the country. So these black lines are coal mining regions. The coal mining industry collapsed somewhere between the 30s and the 80s in the previous century. A lot of joblessness and stress in those areas. It's very different to live in such an area than in the richer areas of the country. And we saw that people that were born there and left had a higher polygenic score, so that's this one, than the entire country on average. And the people that were left behind had the lowest. So there was the detectable brain drain that we could detect with these polygenic scores going on. And something else very interesting that we saw was, we saw that if you compare the birthplace and the current address of these polygenic scores, then you saw the geographic clustering increase. So this is the increase in clustering. But if you look at the geographic clustering of the PC, so those old ancestry differences, those decrease. So those old nice ancestry patterns, those get broken up. And in place of that, the polygenic scores start clustering. Just last week, a preprint came out from the Estonian Biobank, where they looked at like 200,000 people, that's 20% of their population. They did exactly the same analysis as here, and they saw exactly the same thing happen. But then with the polygenic scores were going to the big cities. They have 2 university cities. That's where the higher polygenic score clusters, and those ancestry differences get broken down the same way as in Great Britain. And so this is the longitudinal version of that migration plot. So we broke it down, and you see it like this. So this is from the coal mining region that drops faster than the rest of the country. And this green line here, that's consistently higher, that comes from this brown line. So this, the brain drain is going on consistently throughout the century, and it's increasing the differences. And as I said before, this polygenic score, it's not genes that make you higher educated. It influences a lot of underlying traits that influence these outcomes. And one way to get an idea of those traits is to look at the genetic correlation with educational attainment. So this is the genetic correlation of educational attainment with educational attainment of all of these traits. And this is the geographic clustering. So the more genes they share with educational attainment, the more they cluster. Here's intelligence. Educational attainment has like 0.6 and 1, so that would be here. So it's like pulling all of these traits with it, and not randomly. So as I said, it's very different to live in a poor region of the country than in a rich region. Here's a clearly environmental measure, namely the number of fast food restaurants in a region. It shows pretty much the same map as those polygenic scores. So you have genetic effects and environmental effects clustering together, causing so-called gene-environment correlations. So I'm going to skip a bit fast through this last part. Basically, what we did here is we tried to figure out whether these gene-environment correlations extend beyond the family. We used a couple of sibling models. So here we have a man and a woman living together. They have 2 children. They pass down not only their genetics, but also environmental circumstances. So this is what we call passive gene-environment correlation, because they're just born there. But there are small differences between these siblings that causes one sibling to go to a better neighborhood than the other sibling. And that's what we call active gene-environment correlation. And that's much more difficult to control for with this within sibling designs. So we also controlled these GWASs for where people, so we did more than 50 GWASs on all of these traits, and then looked at what happens if you control for where these people live, if you do a GWAS. And just as a covariate, you have where these people are either born or moved to. We saw the heritability decrease only for traits that are very closely related to socioeconomic status, so education, intelligence, income, time spent watching TV, overall health, age at first birth. But if you look at the change in their genetic correlation with educational attainment, these stars show that almost all of the traits were different. So the heritability doesn't change of the traits, but the content of the signal changes. Most strongly for things related to BMI and substance use, so things that you put into your body. So in other words, if you do a GWAS on BMI, you're capturing things that have to do with the metabolic system or whatever. but you also carry genes because those influence whether you live in a neighborhood with, let's say, a lot of fast food restaurants. If you control for that where people live, then you reduce a little bit. You don't remove it entirely, you just reduce it because where people live or where they move to is only a crude proxy of their social environment. So I think one of the takeaways is that we, as a society, we make these genetic effects stronger because we reward certain propensities with a better environment and punish the lack of those propensities with a worse environment. So if you see that a child is doing well in school at an early age, it's more likely that he gets sent to a better school surrounded with better teachers and other kids that do well. More likely to get a better job later, more likely to live in a neighborhood with better environmental circumstances. And he's also more likely to meet a partner in that neighborhood where he has children with, combining both their genetic and environmental luck in the next generation, which widens the distribution, both like economically, but also genetically, we see. This makes society more unequal over time. And it also makes studying genetics more difficult, especially if we want to do social science genetics. That's why we hired this small army of very smart young people to help us figure all this stuff out. But I think we also need to have some lunch first. But I'm also happy to answer questions if we have time.
I was thinking, you mentioned that you find the resources like the Live Bank and their investment. I think there's one side of them that we may need to be careful about, that's that they're very selective at baseline with very low response rate. How that would influence
Yeah, so they are more likely to be higher educated, more healthy, and female. And we looked at how that influenced our finding in that specific paper, because we also had publicly available data from census data, which is a lot less biased. So obviously, we could not look at the genetic effects, but we could look at the phenotypic outcomes and also on a regional level. And we saw that in the census data, those regional differences were bigger actually than in our data set. And that's because you miss a lot of the lower part of the distribution. So I think, we guessed that probably these effects are bigger than we see. Oh yeah.
Compared to the census data, without first propensity rating, and then we explore the associations and see the rate change, and we also explore V-lots and V-lots effects. So you can validate that for the strange population and the sample. The sample about kind of regions around cities. So it's quite balanced.
That's something I think that probably ways we also work. It's such a small collective proportion of the entire that you can still work properly.
Yeah, decently. What we do is we find the original sampling population at the one city, but it's also I think we then that I need to do that. So basically, we correct technology when that population on 5% is selected to PPD, so there's the higher education, health biases because of the selection, people are born here. But I think if you compare to the entire UK, the differences out of this correction, you still get close to the UK. So, yeah. Yeah.
Good question. Thank you. Oh, there's another one.
I was wondering what's the risk of uniting 100 principal components to kind of work on population
Yeah. Yeah, so usually people use 10 or 20, but we were looking at geography, and we know that ancestry is really strongly correlated with geography, so we wanted to be extra stringent. We actually did see that after 25, it didn't matter that much, but we threw in the rest just to be safe. Yeah. It really depends on the data set. Like the bigger your data set is, the more principal components you will have that pick up that ancestry, because you will have more power, your principal component analysis. It varies a lot between the data sets, and I just would really recommend to try and understand what these principal components are capturing, because they're not magical ancestry capturing approach. It's just captures the biggest patterns in the data set, and if you have like quality differences, it will capture that first and miss probably a lot of the ancestry differences. So I would recommend to put in a lot of effort in computing those principal components, finding out what they are capturing, and so you can control appropriately for confounders.
Quality of the gene typing?
Yes. Yeah, if there are quality differences, that will be picked up probably better than the ancestry differences. Yeah. I found out through through a hardship. Any more questions? No?
So we're all going to lunch now, so you can just follow teachers. You know where the restaurant is. Only one restaurant. There's only one restaurant. So yeah. And remember the badge. Yeah. I haven't asked, but I don't know. Yeah. I think I'm gonna leave it, to be honest. But yeah, I don't know. I haven't asked, and there doesn't seem to be keys on the room, so it's a test of your risk aversion. We're gonna measure it, and we're gonna see.